Sorry for being late and starting the class. I made the mistake of checking my email, and I had so many emails to answer that it just took me all the time. In fact, I've got one email I have to answer that I haven't figured out even how to answer it. So there's one more I've still got to do. Anyways, are there any questions before we begin? Not hearing anything, so let's talk a little bit about the syllabus. So today, this lab is from 7 to 8:50. Um, I'm not sure why there's so much confusion on when the labs are and when the lecture is and what the links are for the Zoom session. I emailed that to everyone, so that's why I'm not certain why there's so much confusion. For people who are using the catalog, I'm just going to tell you for this term, stop using the catalog, not the catalog, the calendar. The calendar I know for the Monday, Wednesday class is screwed up. And I don't know about the Tuesday, Thursday, if it's working or not, but just don't use it. Use the Zoom uh, invite I sent you to log into the class. And I did show you another way to log into the class. And I can show that again. Let me get rid of this. Come to your Zoom meeting link in Canvas. It'll be the first page. And it doesn't show here. I don't know why it doesn't. But if you click on all my Zoom meetings, there it is there. And then you'll probably have a join button. I've got a start button. And I don't know why I've got a start button because I already pushed the start button. That's how I started this class. Any questions about any of that?
I know there were a couple people that were on the wrong link again, because I was one of those people. Um, I might
What,
just pop back. Actually,
what
I don't know
link
if I
did you
can.
use?
Well, it was really weird because I used the one on the that I downloaded onto my Microsoft Outlook calendar, and it wasn't working at first, but and this happened to me during the lecture too. But then when I exited and then tried it again a few minutes after class started, it worked. So I don't
Okay,
know.
I wouldn't use a link you get from Microsoft Outlook, okay?
Yeah,
Use the Canvas link or the one I emailed to you.
yeah.
All right, let's talk about, actually, I got to talk about the syllabus first. Um, on the lab material, there is no lab book. Everything will be online. I've already talked about the grading in the lecture, so I'm not going to go over that again. We're not going to talk about microscope use until we actually use a virtual microscope. We will not be handling a real microscope. Safety information for students and physicians. This is set up here mostly for students who are coming into a real microbiology lab because they would be exposed to real microorganisms. You guys, this information is not as important because you will not be exposed to real organisms. We're going to be doing everything online. However, you do need to know about this information because it's important for your uh, education, your purpose, educational purposes as a nurse or a dental hygienist or whatever in the health science field, and you may be quizzed on these questions, on these topics. So you should know this information. The microbiology labs are operated at Clark under a biosafety level 2, BSL2 level, where personal protective equipment is used, meaning all students have to wear a lab coat, have gloves on, and if they're handling a splash hazard, they also must wear goggles. They must also have closed-toed shoes to come into the microbiology lab. So access to the lab is restricted. Only students who are registered for the class can come into the lab. Students who are immunocompromised or living with or caring for an immunocompromised individual, or students who are pregnant or may become pregnant should contact me and their physician to determine their appropriate level of risk. And generally, what happens under those circumstances is I would give them an organism which is very, very low risk, if no risk, meaning it's an organism that does not cause human disease. These organisms are the organisms we do have. If you were to come into a real microbiology lab at Clark, obviously we're going to be doing this online. And so we will just use these organisms and say that we're working with them. But in reality, everything's going to be online and you will not be handling any organism. But we do have biosafety level 1 organisms, which are mostly used, like ACA, if you've heard of that, E. coli, I'm sure everyone's heard of, Pseudomonas fluorescens, Serratia marsens, probably never heard of that, Micrococcus luteus, Staphylococcus epidermidis, and some of the others you may not have heard of. Biosafety level 2 organisms, we do have a few of those in the microbiology lab. You may have heard of Pseudomonas aeruginosa, Shigella dysentery that causes Shigella, I'm sure everyone's heard of. Staphylococcus aureus, I'm sure everyone's heard of. Streptococcus pneumoniae and Streptococcus pyogenes, you've probably all heard of. Any questions about any of that? For those of you who are joining the lab late, you're going to have to view what you missed online. Hopefully I talked about words of advice and comfort in the lecture. Lab safety rules, you do need to follow the lab safety rules in the microbiology lab. Although we're not going to be physically meeting in a microbiology lab, learning lab safety protocols is a required component of this course. And many nursing programs report that safety is one of the main skills that they want their students to learn. So safety is important, and never take shortcuts which risk safety and accuracy in the lab. All right. The first thing, you're not allowed to take personal items to your lab bench, with one exception. And anything that's personal would be confiscated. It would not be returned to you until it's been sterilized by putting it in a bucket of bleach for 15 minutes or autoclaving, also for 15 minutes, which would destroy anything that's electronics. The exception is you can take personal items that you are personally wearing, such as glasses, a watch. For someone who's got hearing aids, you could take those into the lab. Jewelry that you're wearing. The only request is that you do not touch your glasses or hearing aids or your watch while you're in the lab. And if you're wearing a watch, it should be underneath the glove that you're wearing. All other equipment that you need, like paper, pens, pencils, will be provided for you. And when you use them in the lab, all of that stuff must remain in the lab. None of it can leave the lab. So if you're writing your notes in the lab and you want those notes, you may not take them home. What you can do is take a picture of it, email it to yourself, and then you can use your notes at home. Any questions about that? All personal items that you bring for the microbiology lab, I'm not saying bring into the microbiology lab, you store it in the cabinets that I call cubby holes, which are by the door. The door of the room that we have for microbiology and the cubby holes we consider outside the microbiology lab. So you put that stuff in the cubby hole, and then you just walk a few feet to the sink, and that's where we call the lab proper, just a few feet away from the door or the cubby hole. And that's when we say you've actually entered the microbiology lab. So if you were to use the phone in the microbiology lab, I'm talking about your personal phone, what you'd have to do is take off your gloves, take off the lab coat, go wash your hands, go answer the phone, actually go grab the phone, take the phone outside the lab, outside the room of the lab, and then use the phone. And then when you're done with that phone, you obviously hang up and then bring it in the lab, put it back in your backpack or in your cubby hole, walk over to the sink, and that's when we're saying you walk into the lab at that point, the lab proper, wash your hands, put on gloves, excuse me, put on your lab coat, put on your gloves, and then return to your workspace. Now, it is true that if for some reason you did need to use the phone, we do have a landline in the lab, and you may use that phone even with your gloves. And so that one can be used in the lab. But other than that, your personal phone has to be used as I mentioned. Anytime you were to use something in the lab or were to have it in the lab, personal property would be confiscated and would not be returned to you until it had been sterilized, even if the sterilization would destroy it, like your cell phone could not survive the sterilization. You should consider all cultures you could be working with in the real microbiology lab, potentially pathogenic. Keep hands, pencils, and other materials out of your mouth, your ears, your nose, etc. No eating or drinking, smoking, gum chewing, or any oral habit is allowed in the lab. You will be wearing gloves in the lab. You should monitor yourself for possible symptoms of infection like diarrhea, fever, abdominal cramps, and call your healthcare provider if you notice any of those symptoms. Now, obviously, you won't have to worry about that because we're going to be doing everything online. We have a document on the Canvas website entitled Information for Students and Physicians. And you should use that if you were actually going into a microbiology lab. Coming into the lab, you must wear proper laboratory attire. Students must have closed-toed shoes worn at all times in the lab. In the summertime, if you had something like sandals or women's open-toed shoes, you must change your shoes before coming into the lab proper, either before you enter the room of the lab or before you walk over to the sink to wash your hands. Lab coats are be required and button at all times whenever you're in the lab. We do supply lab coats, although you can purchase one from the college bookstore. Once you bring it into the lab, it stays in the lab for the entire term. Never it will be left. If you want it back, you simply turn it into your instructor at the end of the lab. I would then turn it into the lab people. They would autoclave it and then wash it and then dry it and hang it up and then you could pick it up after the lab has ended, meaning after the term is ended. You could pick it up. Let me think about that. You might be able to pick it up on the first day of the new term, but you should really pick it up between terms. Goggles are required to be used anytime you're working with a splash hazard that includes a liquid culture having organisms in it or something that could splash into your eyes and harm your eyes. Once you no longer have the splash hazard, you may remove your goggles. Cleanliness is a virtue in microbiology, so you should disinfect your lab bench area before you begin. And then when you're all done, after you leave, before you leave, you should disinfect your lab bench once again with disinfectant. You should also wash your hands before you come over to your workstation. And then you should also wash your hands before you leave the lab. Accidents should be reported to your instructor. Minor accidents should be cleaned up as follows. Always clean broken glass first. If there's no bacteria around the broken glass, you pick up the broken glass, and what you do is you use a dust pan and a dust broom to pick up the broken glass. Never pick it up with your fingers because we don't want cuts in a microbiology lab, especially when there's microorganisms with the broken glass. Dispose the broken glass in the broken glass uh, receptacle, the broken glass container. If there's a spilled strong acid or base, which shouldn't happen because we don't have those in the microbiology lab, but it should be diluted and then wiped up. Spilled bacterial culture should be cleaned as follows. This includes if there's broken glass in the spilled culture. Cover the spilled material with paper towels, drench the paper towels with disinfectant, let the disinfectant sit for 15 minutes before wiping it up. Use extra paper towels to wipe up the liquids. If there's broken glass, pick it up first and put it in the broken glass container after the 15 minutes, and then wipe up the area with dry paper towels and discard all the paper towels in the biohazard trash can. Never put glass in the biohazard trash can. Any question about any of that? All right. Be sure to keep your hair and paper away from the Bunsen burners and back to incinerators. If you have long hair, you should tie your hair when you first come into the lab. Gas and bacto incinerators should be turned off before you leave the lab, and really they should be turned off whenever you're not using them in the lab, or will not go back to using them within a few minutes when you're in the lab. Like if you're going to not use them for a half an hour, you should turn them off. And then just turn them on before you use them again. Mouth pipetting is never allowed in the microbiology lab. Never take cultures out of the lab. Samples can only be brought into the lab once you clear it with your instructor and get permission to bring them into the lab. If you have a cut or a sore on your hand, you should wash your hands normally. Then go to the first aid kit, grab a Band-Aid, put on the Band-Aid over the cut or the wound, then put gloves over the Band-Aid. And then when you leave, take off your gloves, go wash your hands. You may, if you desire, put on a fresh Band-Aid, and then you may leave. Any question about any of that? All cultures in the microbiology lab must be completely labeled with your name, the date, the nature of the specimen. For example, if it's E. coli, just write E. coli. If it's something that you don't know what it is and you got it like, say, example, exposure to the air, just write air exposure. And then it should have the medium on the container as well. And then the temperature is optional. That may be added. Old specimens must be discarded properly in the following way. Agar plates are put in a plastic container, and so those agar plates will be discarded in the biohazard container. Paper that has bacteria contamination should also be put in the biohazard container. Plastic pipettes that have bacterial contamination should be put in the biohazard container. Anything that's glass, even if it has bacterial contamination, does not go in the biohazard container. Something that's glass should go in its proper discarding place. Glass slides should go in the glass slide container, which has bleach or disinfectant in it. Cotton swabs should go in the cotton swab containers. Cover slips should go in the cover slip container. The distinction, which I didn't mention the other two labs, is we will wash, well, the disinfectant sterilizes the glass slides, but then we wash the glass slides and we'll reuse them. The cover slips we do not reuse. So they will be put in the disinfectant and then eventually put in the broken glass container. We do use glass test tubes in the lab, and they should be put in the test tube rack for being discarded. The test tubes will be autoclaved, and then they'll be washed and then dried, and then will be reused. We don't really use beakers and flask in the microbiology lab, but in the rare occasion when somebody would be doing something special and would be using it, it would be put next to the glass test tubes for discarding. Is that everything? Oh, any plastic pipettes, anything plastic that does not have bacterial contamination, anything paper that does not have bacterial contamination should be disposed of in the regular trash can. Any question about any of that? Okay, the regular trash can will just be put in the trash can outside the building and would be picked up and probably put in the landfill. The biohazard container will be taken and then autoclaved before it would be put in the regular trash. Any question about that? The autoclaving, of course, would destroy the bacteria. Supplies needed for the lab, for that lab, will be at the front of the lab. Stains and cultures, like media, would be in the back of the lab on shelves. And when you're discarding the slides, the test tubes, what else? I guess that's just, oh, the pipettes would be in the back of the lab. The swabs could be in the back of the lab. We usually do have one container in the back of the lab, but for the swabs, we usually have a container on each of the combined lab benches, meaning your lab area would be connected to the next person's lab area, which would be connected to at least one other area, if not four areas, and there will be one swab container on each side of the lab benches. You would have a signed drawer where you may store materials. You would also have paper and pencils in those drawers. And the paper and pencils are for your use in the lab. None of that material should ever leave the lab unless it has been sterilized before it leaves the lab. So remember, the place where we put our books, our backpacks, our coats is considered outside of the microbiology lab. And then you walk from that area and the door into the microbiology lab proper. Any question about that? All right, you must follow those rules while you're in a real microbiology lab. We would actually have you sign that you would sign your name, that you would follow these rules. Obviously, we're in an online lab, and so if you follow those rules, it's not as important, and so we do not have you sign for that. All right, any question about the syllabus and the safety rules? All right, I'm not hearing anything. Let me stop the share to make sure I got students there. 20 people are there. All right. So in the bottom page of the syllabus, I have the laboratory schedule. It's in the third column of the schedule. The first column being the week. We're going to talk about today Lab 00, lab safety training, and you'll be turning in worksheet for Lab 00. It must be turned in by 8 a.m. Friday, January 12th, or you will be dropped from the course. This will be the only time I take attendance for the entire class, the lecture and the lab. All right, let's go to lab module 00. You're not going to be working in the lab, but you do need to know this material, as many of these rules are relevant for your career, as well as the fact you will be tested on this material. The learning objectives for this lab: 1, be able to distinguish between safe and unsafe laboratory practices. 2, understand where relevant safety equipment is located in the lab. You don't need to know where the safety equipment will be in the lab, because you're not ever actually going to go into a real microbiology lab, but you do need to know what safety equipment is present in the microbiology lab. And then 3, understand how to safely dispose of contaminated materials. So microbiologists have to observe a number of safety rules before they can work with potentially dangerous microorganisms in a microbiology lab. You're not going to be performing any of these activities because everything we do will be done online, but you must understand the guidelines and the rules to create a safe laboratory environment. 1, you must wear proper attire, which includes a clean lab coat covering your lap. It'll be buttoned, disposable gloves, closed-toed shoes, and goggles when a splash hazard exists. The lab coats, the goggles, and the gloves are supplied in the lab, and you put them on after you have entered the lab, and they must be taken off before you leave the lab. The gloves, if they have no bacteria on them, are just put in the trash can. If you have bacteria on your gloves, you should dispose of them in the biohazard container. The goggles should be put in the goggle place, and they will be sterilized with UV light for the next student's use. And then the lab coat should be hung up in the lab, ready for your use next time. They would not be washed, well, actually autoclaved first, and then washed until the end of the term, meaning after the term ends, that's when they would do that. And then the students at the start of the next term would have a fresh, clean lab coat to work with. Any question about any of that? All right, you must wear closed-toed shoes while you're in the lab, gloves, and if you're going to be working with a splash hazard, you must wear goggles while you have a splash hazard. Long hair should be tied back if you're coming into the lab. Absolutely no food or drink at any time in the lab. No use of electronics. And really, unless you're wearing the personal belongings, you should not bring personal belongings into the lab. If you do, any personal belonging would be confiscated and you would not get it back until it had been sterilized, which would destroy a cell phone. So remember, the personal items are stored in the cabinets next to the door. The door and the cabinets, what I call cubby holes, is considered outside the lab proper. And you have to walk after that space a few feet to get into the lab proper. All right, this is showing you some of the things that people wear for safety when coming into the lab. We've mentioned boots or closed-toed shoes, hair tied back, lab coat, gloves, and if those are goggles if there's a splash hazard. Those might be the person's eyeglasses, which you are allowed to bring into the lab. If you do bring eyeglasses into the lab, you should not touch it with your gloved hands in case you have bacteria on your hands. And the same with jewelry. I don't see any jewelry on this person, but if she were wearing jewelry, jewelry may be brought into the lab. Another thing which is shown is pants should be brought into the lab, and then a shirt or a blouse, which you can't see there, but keep your work area clean and uncluttered. You should disinfect the lab station where you work before you begin the work in the lab, so there's no microorganisms around. And then you should disinfect the area before you leave to kill off any microbes that may have spilled onto the lab bench, so it's clean for the next student who comes along. This is an actual picture of our work area in the microbiology lab at Clark. Any questions? All right, let me go over where you discard waste in the lab. Regular trash, which would be things like paper, plastic pipettes, anything that's not sharp, that's paper or plastic, goes in the regular trash can. That would be gloves that are not contaminated with bacteria as well. Anything that's sharp, like a scalpel, which we don't use in microbiology, a razor blade, which we also don't use in microbiology, a needle, which we do not use. And then anything that's glass does not go in the trash can. The same with the biohazard container. No sharps in the biohazard container, no glass in the biohazard container. The reason for this is that the people handling the garbage do not expect glass or sharps, and we don't want them to get injured handling the garbage. Contaminated material, meaning material contaminated with bacteria, should go in the biohazard container, assuming it's not glass or a sharp. And that would be plastic, like a plastic pipette that's contaminated with bacteria, any paper that's contaminated with bacteria. The Petri dishes are plastic. And when you're all done with them, you should put them, if they have bacteria in them, you should put them in the biohazard container. If you had a empty plastic container that did not have contamination with bacteria, that plastic Petri dish could be put in the regular trash can. Generally, you won't have that. Because if you're using it, you're going to be having bacteria. All right, test tube bends go in the test tube rack for disposing. That's in the back of the lab. We don't really use beakers or flasks, but they would be next to the test tubes if you were to use them. And then we have special containers marked for swabs, contaminated swabs, contaminated slides, and then contaminated cover slips. You should report all accidents immediately, no matter how slight. Cover the material with paper and then drench the paper with disinfectant, allowing the disinfectant to soak for 15 minutes, and then clean it up. Since it had bacteria, it should go in the biohazard container. Never pour reagents back into a container. Absolutely no mouth pipetting in the microbiology lab. No chewing your fingers, no chewing a pencil, no oral habits like chewing gum, no smoking. A Bunsen burner, and then back to incinerator. The purpose of these is to sterilize your tool before handling microorganisms. So I don't know if that's a loop or not. I can't see it, but this is a loop. And they're putting it in a back to incinerator, which you can see is red hot. It does take about 5 minutes for this to get red hot. And what they're doing is they're sterilizing the loop. You can see the loop is turning red. You should just hold it in there until the loop turns red and then pull it out before using it. Generally, you cool the loop before you use it. Like if you want to pick up cells, you don't have a red hot loop or a real hot loop to pick up cells. You either cool it for 30 seconds, holding it up in the air, or more likely, you touch it against something that's sterile to cool the loop, and then go and pick up your cells. All right, so this is the safety equipment that we have in the lab. The fire alarm right there. The eye wash station, scroll down right there. The fire blanket right there. The fire alarm right there. Fire extinguisher right there. Fire blanket right here. You can see a little bit of it right there. The first aid kit right there. And then a chemical shower right there. In all the years I've been teaching, and in all the years I've actually also done research in a lab, including a microbiology lab, I've never used any of these except for the first aid kit, meaning I've given students bandages. I've never used anything else, but I have given students bandages. I have in the labs I did research in seen someone use an eye wash station. They spilled the chemical in their eye and they had to wash it out in the eye wash station. I've never had to use or seen the use of a fire extinguisher in the lab, use a fire blanket, pull the fire alarm, or use the shower. If you do use the shower, you should know that this actually, the water goes on the floor. And it is true we have a, what do you call that, a drain on the floor, but it will make a mess to the floor because of the water on the floor. All right, you should pre-read relevant labs before coming to the class so you can get the work done right away. It's not as important in an online lab, but you should be familiar with the day's procedure, so it's best to pre-read. All right, this goes through the lab safety rules again. I won't go through most of it because I've already gone through most of it, but I will cover things that I have not. But you need to know about safety because it's important for your career. In the nursing program, they really want their nursing students to understand safety, especially coming out of the microbiology lab. And you also will be quizzed on this material. Just looking to see what I haven't talked about. Oops, too far. So no one comes into the microbiology lab except for college staff and students who are registered for the class. No personal items come into the lab proper. You should consider all cultures you're working with to be potentially pathogenic. That's why we require you to wear gloves, why you can't eat or drink, smoke, chew gum, or any other oral habit, chew your pencil, chew your nails in the microbiology lab. You should monitor yourself for possible infection with a bacteria. That's not important for you guys, but if you were to go into a real microbiology lab and then were to get diarrhea, fever, or abdominal cramps, that's a possible sign of getting contamination or infection of a microorganism found in the lab. And then call your health provider. And then we have a document information for students and physicians to provide information to you and your physician. All right, you must wear proper attire. Let me just go over that once again. And obviously you have to have closed-toed shoes, lab coats, and gloves. And if there's a splash hazard, you must wear goggles. Accidents have to be reported to your instructor, no matter how small, but it is your responsibility to clean it up. I'm not going to talk about this because I already did. Be sure to keep your hair and paper away from Bunsen burners or back to incinerators. If you have long hair, you should put on a tie as soon as you enter the lab or before. Gas and back to incinerators should be turned off before you leave the lab, and really they should be turned off when you're not using them. No cultures may ever be taken out of the lab. Samples can only be brought into the lab once you get permission to do so from your instructor. All microbiology cultures must be completely labeled with your name, the date, the nature of the organism, and the medium used. Optionally, you may include the temperature that you grow the organisms at. I already talked about disposal. And supplies are provided for you at the front of the lab. Generally, when you're turning in your used pipettes, your used test tubes, you usually turn them in at the back of the lab. All right, any questions about any of this lab?
Hi, can I ask a question really quick? This is Katie. I just noticed on number 2, it talks about personal items, and it says on letter C, it says that cell phone usage is prohibited, but you had mentioned that we could take a picture of notes. And so does that mean that we, like, in theory, we could bring a cell phone to take a picture of our notes?
No,
Or okay.
we
So
have a iPad or a notebook that remains in the lab and stays in the lab, and you can take a picture using that.
I get it. Okay. Thank you.
All right. All right, I think that's it. Let's go to the worksheet. So laboratory worksheet 0, or to lab module 00. You need to answer these questions. Answer each question for each asterisk. Like rule 1 has 4 asterisks. That means there are 4 answers to rule 1. In the lab, what do you always wear for safety before you enter the lab? This is not directly stated in the lab, but it is shown in the picture I showed you of the lady. Where is it?
Closed-toed shoes.
Thank you. I was going to ask what's one thing. And this picture does show you what you need to be wearing in the lab. And these items must be put on before you come into the lab. All right. So what's an example of something that you put on after you enter the lab?
A clean lab coat that covers your lab while sitting.
Yes. All right. So that's one example for rule 1 and rule 2. The important thing is to note that rule 1 is something you put on before you enter the lab. Rule 2 is something you put on after you enter the lab. We're talking about the lab proper, by the way. And then rule 3, you need to look up what a safety data sheet is online and what's in a safety data sheet. We have a safety data sheet for each chemical in the lab, including something like sodium chloride. Provide at least a phrase for each answer, meaning a one-word answer is unacceptable. So you need to describe what the safety data sheet says about a chemical. And you need 5 answers here. Any questions? Rule 4, know your safety equipment. Name 5 items of safety equipment found in a microbiology lab. Rule 5, name 2 items that you can take into the lab. And those are personal items or something like that. There are some things you can take in the lab. And then can you eat or drink in the lab? Just answer that yes or no, and that's 3 items under Rule 5. And Rule 2, list 1 item of laboratory attire that you must wear in a microbiology lab. And this could be a repeat of Rule 2. Or it could be a repeat of Rule 1 too, because it doesn't say when you must put it on. So it could be a repeat of Rule 1 as well. And then list number 3, list 1 item belonging to you that is allowed to be brought into a microbiology lab. And then 4, list 1 item belonging to you that is not allowed in the proper microbiology lab. So answer these questions and then submit the file by 8 a.m. Friday of week 1 in the folder for Worksheet Lab 00. If you do not, then you may be dropped from the course. Generally, I'll go in there if I haven't heard from the students and they don't turn in the first lab, I just drop them from the course. Any questions about any of that? All right, if there's no questions, I'm going to stop talking and I'll be here to answer any questions until 8:20 or until the last student logs out.
I actually have the lab 00.
Uh-huh.
For the instructions, it says fill out this worksheet. So do we have to download it or do we do it on a separate doc and turn it in?
You may do either.
Okay, thank
Okay.
you.
So if you want to type it, you may type the answer. However, I would request that you put in the question. So add the question and you can just copy and paste it into a document. And then of course answer it. All right, No other questions? Let's begin work on the lab. Like I said, I'll be here at least until 8:20, longer if the lab goes to 8:50. I gotta look and see.
Would now be an okay time to ask a quick question about the Zoom links, or should I ask that at 8:20?
You can ask that now. I'm done talking.
Okay, so I just
Just
wanted
a
to,
minute, just a minute. I do want to
okay,
say I will be here until 8:50 or until the last student logs out to answer questions. All right. So what's your question again about the
yeah,
link?
so yeah, my question is just that I went ahead and I tried to access the Zoom link from the tab in Canvas that says Zoom meetings. And then I noticed that on your screen when you clicked on Zoom meetings, um, there was like an additional, blue hyperlink that you were able to click on that said something like all Zoom meetings or something. And my Canvas doesn't have that. And so I don't know if
It has
it's,
this, doesn't have this link?
oh, you're not sharing.
Oh, sorry, just a
Yeah.
minute. It doesn't have this link
Correct. Yeah. I don't have the thing
down
that
here?
says nope, nothing.
Is there
Um,
another link you can click on?
no, the only thing that I have is the thing that says get training. And then I have the three different tabs that says upcoming meetings and previous meetings and cloud recordings. I don't know if anyone else is experiencing that. I'm using the Microsoft Edge browser. I don't know if maybe it's my browser. I could try a different one.
No, I don't have anything under these either.
So maybe I don't know, is that something that maybe I should talk to the IT people about?
They'll probably tell you that what I need to do is schedule the meeting using this link because
Mm-hmm.
these meetings I schedule directly in Zoom. The reason is that I have more options to choose. And certainly one of the things I can do from Zoom is I can mute everyone. So when you guys log in, you'll be muted. And I can do that if I schedule the meeting in Zoom. If I schedule it here,
Mm,
I don't have that option.
I see.
So I have fewer options. There's one other thing that I also add, and I don't remember what it is, but that one I did remember.
Oh, it looks like other people are saying that they also have the same problem in the chat. There's a couple other people that said they
Yeah,
have
you
that
don't have
same.
this place to get. I looked into doing it here, and I was wondering if I could like schedule a meeting and just put in an old meeting.
Mm-hmm.
I'll look into doing that again, but I didn't see how to
Okay.
even do that.
Yeah, and
Um,
somehow I don't know. I've been confused tonight with the Zoom meetings because it was like when I tried the link the first time it wasn't working, and then I exited the meeting and then I came back in a few minutes later and then it was working. But I've been able to get here. It's just been kind of strange.
I think the problem is, are you the one who's using Outlook to do it?
Well, yeah, but I got the links from Canvas because there was like an option to upload
The document.
the the Zoom the Zoom meetings into your calendar. And so
All right,
I did
I
that.
told you don't use that. Don't use the calendar this term. Okay, so don't
Okay,
use
but if I
this.
don't have that and I can't use this Zoom.
Emailed you a link
Oh, yes,
to
I
the
did
Zoom.
get that, but
Okay.
now was that email just but that was just for the lecture, wasn't it?
No, it was both for the lecture and the lab.
Oh, now,
I sent you two.
you know what? I'm sorry. When you were saying email, I wasn't thinking my personal email. I was thinking like the Canvas email.
I'm not sure where I sent it, so you have to look for that.
Okay,
Another place you can
I
get
don't see it anywhere.
in Canvas.
I don't see it in my personal email or in I did no, sorry, I did get one in Canvas. Let me look again.
It looks like the one in Canvas, professor only has the lecture
Yeah. Yeah.
link
And
in
that was
the
sent
if you
at 5:30, 5:35 a.m. on January 7th. And that was
if
the
you
Just a minute, let me try and get there.
If you go into Canvas on the homepage, underneath the
Yeah, I've got
course
it
information.
here too, but um, I thought I emailed that to everyone. Let me see if I didn't send that. Now let's go down quite a ways. Of course, I might have sent it through CTC link also. It looks like from on Canvas, I've only sent the lecture to the Tuesday, Thursday people. Let me scroll down further. But I might have sent it in CTC. How do I get there? Nope, I'm not going to get there from there.
And just so you know, Professor, when I said that I was using my calendar, I wasn't using the Canvas calendar. I was using, so the links that I was using were the links that came from the Canvas where it says Zoom Meet invite, and then it has Zoom Meet invite lecture Tuesday, Thursday, and then lab Tuesday, Thursday, 7. And so because there was like a Zoom meeting link connected to that document. And so, and that just, I ended up importing that into my personal calendar just so that I would have it on there. But that was the link that was acting kind of funny. Like it was working at one point and then it wasn't working.
Yeah, I would say don't use the Outlook.
Okay. Okay.
All right, let me look here. I might have sent it here. The trouble is I'm not sure I can look here to see if I've sent something. Oh, fudge. Yeah, I don't see how it's Monday, Wednesday.
I think it's easier if you just go into the modules on
Yeah,
Canvas
I'm
and
going to
go
just say
through
there
the.
that I've got the invites up here as well. So there's the lecture for Tuesday, Thursday. There's the 3 p.m. lab for Tuesday, Thursday. There's the 7 p.m. lab for Tuesday, Thursday.
Yeah, those were the ones that I tried to use, but again, I uploaded them into Outlook, so
Outlook,
maybe I'll
yeah.
just not do that
Yeah,
next time and I'll just
you can
try to.
download the page directly. I don't know if it can work for inside a Canvas.
That's what I did though. Well, actually I don't know if it's this, I don't know if it's the same thing if I download.
Is that it there?
Go into that page and copy and paste the link that's down underneath that says join the Zoom.
Yeah.
Meeting right there, if you literally just copy and paste that into your browser
Yeah.
and hit like enter, it'll bring you to it and it'll ask you to join the meeting and you can do all that from there.
Okay, perfect. Well, if all else fails, I'll definitely do that next time. Thank you.
That's what I did as well, and it worked for
Okay.
me. I was able to just go to the Zoom link from the modules and I just copied it and then hit search, and it just took me to that Zoom page, and that's how I got here, and it worked.
Oh, perfect. Okay. Thanks for everyone's input.
All right. Is this recording good? Anyways, if I didn't send you the lab invite, I apologize. I thought I had sent everybody the lab invite, but you can get it from your homepage. For Tuesday, Thursday, the lab right here or there. Okay, I'm going to stop recording now, stop share. And then you guys can work on the lab. I'll be here for at least 20 more minutes, maybe 50 minutes. I will say that once the last student leaves, I'm going to log out as well. But I'm here to answer any questions.